Lipids are chemically diverse group of organic compounds, insoluble or poorly soluble in water, but readily soluble in non-polar solvents such as ether, chloroform, benzene. These are not polymers like proteins, nucleic acids, and polysaccharides. The hydrophobic nature of lipids is due to the predominance of hydrocarbon chains in their structures. Lipids have diverse functions. For example, storage lipids are the source of metabolic fuel. Structural lipids provide the structural components of membranes. Some have protective functions in bacteria, plants, insects, and vertebrates, where they make coating between the body of the organism and the environment. Some are pigments like keratin, hormones like vitamin D derivatives, sex hormones, signaling molecules like ecosonoids, cofactors like vitamin K, detergents, that is bile salt, and many other specialized functions. Fatty acids are the simplest form of lipids. These are long chain hydrocarbons, around 4 to 36 carbons long, with one carboxyl group. In the biological systems, fatty acids contain an even number of carbon atoms. The 16 and 18 carbon fatty acids are the most common fatty acids found in the biological systems. Fatty acids are amphipathic in nature, that is, they have both non-polar and polar ends. The alkyl chain may be saturated or unsaturated. Unsaturated fatty acids contain one or more double bonds and called monounsaturated or polyunsaturated respectively. The double bonds in naturally occurring fatty acids are generally in cis configuration as opposed to trans configuration. And in polyunsaturated fatty acids, these double bonds are almost never conjugated. That is, alternating single and double bonds are present. Mammals lack the enzymes to introduce double bonds at carbon atoms beyond carbon 9 in the fatty acid chain. Therefore, mammals cannot synthesize two fatty acids, that is, linoleate and linolenate. Both are therefore essential fatty acids, that is, they must be obtained from the diet because they are required by an organism but cannot be endogenously synthesized. The melting point of fatty acids depend on chain length and degree of unsaturation. The longer the chain length, the higher the melting point, and the greater the number of double bonds, lower the melting point. Double bonds make unsaturated chain more rigid. That is, unsaturated chains cannot pack themselves in crystals efficiently and densely as saturated chain. So they have lower melting point as compared to saturated fatty acids. Similarly, the unsaturated fatty acids with cis configuration have lower melting points than the unsaturated fatty acids with trans configuration. Triacyl glycerols or triglycerides are triesters of fatty acids and glycerol. They are composed of three fatty acids and a glycerol molecule. These are of two types, simple and mixed type. Those containing a single kind of fatty acids are called simple triacyl glycerols, and those with two or more different kinds of fatty acids are called mixed triacyl glycerols. These are nonpolar, hydrophobic in nature, and a major form of stored lipids. Because triglycerides have no charge, they are sometimes referred to as neutral lipids. The fatty acids that they contain are of varying length and which may be saturated or unsaturated. These can be distinguished as fat and oil on the basis of physical state at room temperature. Fats are solid at room temperature, contain a large proportion of saturated fatty acids, but oils are liquid at room temperature because of their relatively high unsaturated fatty acid content. Hydrolysis of triacylglycerols with alkalis like sodium hydroxide and potassium hydroxide is known as saponification. Saponification yields salts of free fatty acids known as soap and glycerol. Wax are esters of fatty acids, saturated or unsaturated, and long chain alcohols, which are high molecular weight monohydroxy alcohols. Waxes are biosynthesized by many plants or animals. The best known animal wax is beeswax. Phospholipids are amphipathic molecules which contain fatty acids, a platform to which fatty acids are attached, a phosphate group, and an alcohol attached to the phosphate. Phospholipids are divided into two types based on the platform on which phospholipids are attached. These are glycerophospholipids and sphingophospholipids. Glycerophospholipids are derived from glycerol, whereas phospholipids derived from sphingosine are called sphingophospholipids. In glycerophospholipids, there is a glycerol molecule, two fatty acids, a phosphate, and an alcohol like choline. These are the most numerous phospholipid molecules found in cell membranes. The hydroxyl groups of carbon 1 and carbon 2 of glycerol are esterified to carboxyl groups of two fatty acid chains, and the carbon 3 hydroxyl group of the glycerol backbone is esterified to phosphoric acid. If no further addition is made, the resulting compound is phosphatidic acid, the simple glycerophospholipid. The other phosphoglycerides are derived from this phosphatidic acid by addition or formation of an ester bond between the phosphate group and the hydroxyl group of one of the several alcohols like serine, ethanol, amine, choline, glycerol, and inositol. In sphingophospholipids, 18 carbon amino alcohol called sphingosine is present instead of glycerol. A fatty acid is joined to a sphingosine via an amide linkage to form a ceramide. Ceramide is the structural parent of all sphingolipids. The addition of phosphorylated alcohol to the carbon 1 position of sphingosine leads to a variety of sphingophospholipid. For example, sphingomyelin, in which phosphocholine group is attached to the carbon 1 hydroxyl. Sphingomyelins are the major constituent in the nervous tissue of higher animals. Glycolipids are lipids containing saccharide groups. The glycosphingolipid is the most important type of glycolipid and constitute the third major class of membrane lipids after glycerophospholipids and sphingophospholipids. In the glycosphingolipid, head group contains one or more sugars connected directly to the hydroxyl group at the carbon 1 of ceramide moiety and do not contain phosphate. Steroids are complex derivatives of triterpenes. Each type of steroid is composed of four fused rings called steroid nucleus. A sterol is a class of steroid which has hydroxyl group at the carbon 3 of the molecule. Cholesterol is an example of sterol and is an essential component in animal cell membranes. Cholesterol also acts as a precursor for the biosynthesis of all steroid hormones and bile salts. It is not found in plants and fungi. The types of sterols that is found in plants and fungi are stigmasterol and ergosterol. Ecosonoids are very potent biological signaling molecules that act as short range messengers. These include prostanoids, for example, prostaglandins, and leukotrienes. These are known as ecosonoids because of their common origin from 20 carbon polyunsaturated fatty acids called ecosonoic acids, mainly arachidonic acid. These are potent hormones and act locally instead of being transported in the blood to act on cells of other tissues or organs.